in a department that not only has a wonderful tradition of research in ecology and evolutionary biology going back to the roots in this country, but also today has a very, very vigorous program. I really am truly honored to give this year's Warren Wagner lecture. I first met Herb 40 years ago at the International Botanical Congress in what was then Leningrad. I just watched him give a tour de force talk to a rapt audience, holding them in the palm of his hand through a signature blend of humor and charm and bounding enthusiasm and a deep knowledge of ferns and the evolutionary questions they pose. I introduced myself and was surprised to find that he already knew who I was, a mere graduate student, based on listening to a talk I had just given the day before. Herb was very kind and thought-provoking in the conversation that followed, and a number of other conversations we've had over the years, and I've held those memories dear. Actually, I had met Herb much earlier in my life, in a way. When as a 14-year-old, I bought the field guide to the ferns, and within minutes, stumbled onto this wonderful diagram inspired by Herb's earlier research on reticulate evolution in spleenworts of the southern Appalachians. It was my first exposure to how scientists could infer relationships and the evolution of leaf form, and it made a very deep impression. Turns out, Herb and I were both inspired by a noted botanist, Edgar T. Wherry, who served as his undergraduate advisor, I believe, and had a major role in convincing me to go into ecology and botany through his work on the rare ferns and orchids and carnivorous plants in the New Jersey pine barrens. And then much more recently, I inspired one of my own graduate students, Emily Sessa, to use modern techniques to test several of Herb's classic hypotheses regarding reticulate evolution in the fern genus Dryopteris. Emily confirmed several of those predictions while contradicting a few. She recovered part of the genome of the extinct perennial species that Herb postulated and has begun a bright and productive career in fern evolution at the University of Florida. So Herb's impact rolls on. So monocots, with 65,000 species in 83 families and 12 orders, and including such groups as the grasses, the sedges, palms, gingers, bananas, orchids, lilies, yams, aroids, and seagrasses, are one of the most diverse, morphologically varied, ecologically successful, and economically important of all plant lineages. Since they arose more than 140 million years ago, monocotyledons have radiated into almost every habitat on Earth. Today, they dominate many terrestrial and aquatic ecosystems, display kaleidoscopic variation in vegetative and floral morphology, provide most of the human diet directly or indirectly, and comprise nearly 25% of all flowering plants. Understanding their origin, phylogeny, and patterns of morphological divergence, geographic diversification, and ecological radiation is thus a major challenge and opportunity for evolutionary biologists. Now, over the past 20 years, molecular systematists have revolutionized our understanding of monocot relationships. Their studies have led to a dramatic reclassification of monocots, triumphs of monocot molecular systematics include recognizing the unity of the commelinids, 5 disparate orders that share many nucleotide substitutions and UV fluorescent compounds in their cell walls. The finding that several genera once placed among the lilies actually belong to 4 different orders, and the recent discovery that highly reduced aquatic plants in the family Hydrotelaceae once placed in the order Poales with grasses, sedges, and their relatives are actually not even monocots, but basal angiosperms related to water lilies. The emerging monocot phylogeny, as of about 8 years ago, placed Acarus, sister to all other monocots, followed by divergence of the orders Elismatales, Petrosavillales, Dioscoriales and Pandinales, Liliales, Asparagales, and the 5 comelidid orders. However, even the most comprehensive study until recently, including 125 placeholders and sequences of 7 genes from the plastid and mitochondrial and nuclear genomes, left many relationships unresolved or weakly supported. Such soft spots, highlighted on the slide by the warm colors, included relationships among the 5 comelidid orders, among the families of the Poales, the Zingiberales, the Dioscoriales, Pandinales, and Elismatales, and among the subfamilies, tribes, and subtribes of the orchids. To develop a better understanding of broad-scale relationships in monocots and a stronger basis for analyzing their evolution, in 2008, our team obtained a $3 million grant from NSF to conduct a 5-year study of monocot phylogenetics and evolution. Our principal aims are to use a phylogenomic approach to develop a fully resolved and strongly supported backbone phylogeny for the monocots, using next-generation sequencing to obtain whole plastid genomes, or plastomes as they're known, each about 150 kilobases in length, and transcriptomes, each several megabases long, and largely from the nuclear genome. And to do so for species stratified across all monocot families. We also have aimed to reassess the age and the classification of critical fossil taxa to develop a new timeline for monocot evolution. And finally, we aimed to use this new set of time-calibrated molecular trees to analyze patterns of adaptive radiation, character state evolution, historical biogeography, and species diversification. Our team, monototal team, includes from left to right Cecile Linné, Jerry Davis, Claude DePathlis, myself as the lead PI, Maria Gandolfo, Sean Graham, Jim Liebensmack, Chris Piers, Dennis Stevenson, and Wendy Zomwaffer, as well as 6 postdocs, 7 graduate students, and a dozen undergrads. To date, we've published over 30 papers and made over 50 presentations at professional meetings. We've met or exceeded most of our goals, producing over 250 plastomes and more than 100 transcriptomes, totaling in all over 300 megabases of DNA sequencing. That's the equivalent of over 200,000 RBCL sequences, which was the unit used in the early days of molecular systematics to resolve relationships among land plants. Today, I want to briefly review some of our key findings regarding monocot phylogeny, and then turn to what we've learned as a result about the evolution and drivers of species diversity in 2 of the largest families of monocots, the Bromeliads in 5th place with roughly 3,300 species, and the orchids, the largest family with over 25,000 species. We first used plastomes from 68 monocots and 15 other angiosperms to resolve 2 of the most challenging questions in higher level monocot systematics, involving relationships among the comelidid orders and among the families of the Poales, the grasses, through the Bromeliads. Based on a maximum likelihood analysis of over 109,000 aligned bases from 81 plastid genes, we found the first strong support for resolution of the ties among the comelinids with, as it turns out, the Poales, sister to the orders Comelinales plus Zingiberales, and the palms in Ericales, sister to the Australian order Dasypogonales. Bootstrap for the first herbaceous clade was 93%, and 86% for the second woody clade. Our data resolved very short branches, as you can see here, at the base of the cotyledons, which is probably why they have been so hard to identify securely in the past using only a few genes. Resolving very short branches requires large amounts of sequence data, and the noise introduced by subsequent mutation makes the job even more challenging for branches that are both short and deep, as they are in the cotyledons. Our plastome data also resolved relationships among all families of Poales with 100% bootstrap support at 29 of the 32 nodes sampled. Ties among these families differed in several respects from those resolved by previous analyses, with Bromeliad sister to all other Poales, then the cattails and the burreeds, then Rhapadiaceae. The three cyperid families, the sedges, the rushes, and Thorniaceae, were sister to the remaining taxa, with the three xyrid families, the yellow-eyed grasses, pipeworts, and aquatic Maiaca, forming a grade at the base of the ladder. Then you had Rustionaceae, Central Lepidaceae from the southern hemisphere, sister to the grasses and their allies, with the small southern hemisphere family, Ecdiocoleaceae, a word this is much larger than most of the plants in that family, a sister to the genus, to the grasses, which are the second largest of the monocot families. It includes over 11,000 species and many of the most important crop plants on Earth. Now, as indicated by the red dots, this new phylogeny implies that wind pollination arose independently in this one group 5 times. Consistent with theory, we found that wind pollination was significantly correlated with open, windy habitats and small, visually inconspicuous unisexual flowers that lack nectar. Open habitats and a local plant abundance favor wind pollination, and the inefficient movement of pollen to stigmas by wind favors animal pollination in locally rare species or in forest understories. For Poales, I would add that local dominance is often achieved via tall stature, vigorous vegetative spread, and adaptation to patchy disturbances by, for example, fire or flooding. Exceptions are the Central Lepidaceae and Ecdiocoleaceae, found on highly infertile sites whose openness favors short stature, and often in windy alpine habitats where reproductive assurance favors wind pollination. Local dominance may also be favored by positive feedback loops, linking grasses and sedges to fire, to grazers, or to fungal endosymbionts. Now, Craig Barrett, a postdoc with Jory Davis and now an assistant professor at West Virginia, has used whole aligned plastomes to obtain a strongly supported backbone phylogeny for palms and their sister order, Dasypogonales. Among the palms, first the climbing rattans, and then the mangrove palm Nipa are sister to all other groups of palms. Barrett and his colleagues have also analyzed relationships among the 8 families of the gingers, the Zingiberales. Consistent with prior work, they found that the ginger families themselves, namely the Zingiberaceae, the Costaceae, the Marantaceae, the Canaceae, form a clade. But contrary to previous studies, their analysis resolved the 4 remaining families as a clade as well. Support for relationships among 3 of those 4 families, however, was quite low. So Craig and colleagues extended their taxon sampling and included both non-coding and coding positions in the analysis, and that greatly increased the support for the new topology. Even all the information in plastic genomes, all 150,000 bases, however, is not enough to resolve all the nodes with 100% bootstrap support. We've gone beyond what you can resolve with chloroplasts alone. And that befits the fact that in Zingiberales, the problematic branches are very short and very deep. This new phylogeny overturns the arrangement of families by Kress and his colleagues based on morphology and limited DNA sequence data implies that petaloid stamens arose independently in Heliconia and the ginger families, both times in association with bee pollination. Roxy Steele and Chris Piers at the University of Missouri have used plastomes to derive strongly supported phylogeny of the higher Asparagales, the largest of the orders of monocots. And their treatment excluded just the orchids and two other small families at the base of the order. Graduate students, here we go, sorry. I hit the wrong button, ladies and gentlemen, sorry. Graduate students, Maribel Gomez and Vivian Lan and Renee Su, together with Sean Graham at the University of British Columbia, have used plastome data to resolve relationships among all families in the Pandinales and Dioscoriales. Both of these orders are sister to each other, and together are sister to the Commelinids plus the Asparagales and the Liliales. They have recovered a strongly supported phylogeny for the Pandinales using the mycoheterotroph, the non-photosynthetic Sciophylla, as a placeholder for an entire mycoheterotrophic, non-photosynthetic family, the Trioridaceae, the members of which rely on fungi rather than photosynthesis as an energy source. Sequencing the plastomes of these mycoheterotrophs it was a major achievement, really, and it documents a nested series of losses following the loss of photosynthetic capacity. Graham's team has also recovered a strongly supported phylogeny for Dioscoriales, including Bromeliad, as a placeholder for the mostly mycoheterotrophic and tropical Bromeliaceae. Members of the mycoheterotrophic Thismiaceae shown here have yet to be added to their analysis. And yes, it is true, those are plants, they're not marine invertebrates. Members of my team at Wisconsin and the UBC team have finalized a phylogeny for Liliales that places Campanema from Tasmania and New Caledonia with the mycoheterotrophic Horsiaceae from South America and Australasia. Both of them are sister to the rest of the members of the order, and that implicates a Antarctic teleconnection between Australia and South America in the origin of the order, and of two other pairs of families within it. Greg Ross of the Graham Lab has derived a plastome tree, including all genera of Elismatales, which is a largely aquatic order, sister to all monocots except for the emergent aquatic Acris. His phylogeny shown here suggests 2 to 3 independent origins of the seagrasses and a secondary loss of marine but not aquatic habitats in Rupia and the pondweeds. Okay, enough of a march through the monocots at high speed now. Let's turn to a few detailed studies. And first, let's turn to a study of phylogeny, historical biogeography, adaptive radiation, and determinants of net species diversification in bromeliads, conducted by myself, Ken Seitzma, Paul Berry, and several collaborators in a consortium that I lead. Bromeliaceae is the 5th largest monocot family with 58 genera and 3,300 species. It is also the largest of 40 angiosperm families found mostly or exclusively in the neotropics. It contains more epiphytic species than any other family of vascular plants except orchids. Bromeliads bear leaves arranged in a distinctive circle or rosette, and in many species, particularly those that grow as epiphytes, these leaves fit together so tightly that they form a tank that can impound rainwater and from which water and nutrients can be absorbed. Uniquely among monocots, bromeliads bear tiny umbrella-like hairs or trichomes on their leaves, and in many epiphytic bromeliads, such trichomes can absorb water and nutrients at high rates. Bromeliads range from the ancient mist-shrouded tepuis of the Guyana Shield to the sun-baked granitic domes of the Brazilian Shield, from cloud forests in the Andes and Central America to the cypress swamps of the southeastern US, and from the frigid Andean Puna to the arid Atacama. Many species possess CAM photosynthesis and take up CO2 at night to minimize water losses. Tank bromeliads often house a great diversity of insects, including some that adversely affect our health, and other animals, including crabs and frogs and salamanders and even snakes. In a single hectare of cloud forest, the tanks of epiphytic bromeliads can impound thousands of liters of rainwater and hundreds of kilos of humus high in the canopy, while providing key sources of food for many primates and birds. A few tank bromeliads are carnivorous, and at least one is known to benefit from the prey captured by spiders that live inside them. Several species are protected or fed by ants. Pollinators include a wide range of insects, as well as hummingbirds, bats, and a few perching birds. In many cases, the whole plant becomes a showy display to attract pollinators. The inflorescences of puya in the high Andes are the most massive of any flowering plants, while those of some Brocchinia and Tillandsia are only a few centimeters tall. Some species have fleshy fruits dispersed by birds or occasionally mammals, while others have wings or plumes and are wind dispersed. Finally, bromeliads contribute a large share of the epiphytic species in neotropical forests and are particularly diverse at mid-elevations. To address several questions regarding bromeliad evolution, we derived a molecular phylogeny using nearly 10,000 aligned bases from 8 rapidly evolving plastid regions for 90 species, representing almost all of the bromeliad genera, and then used that tree to reconstruct patterns of morphological and ecological evolution, historical biogeography, and rates of diversification. Now, based on morphology, bromeliads have long been divided into 3 subfamilies. The Pitcairneoidea, with winged seeds represented in this ceremony by spear-like inflorescences of puya, Tillandsioidea with plumose seeds, represented by the coats of Spanish moss, and Bromelioidea with fleshy fruits, represented by crowns of Guzmania. Our maximum likelihood analysis produced a well-supported bromeliad tree consistent with our new classification involving 8 subfamilies, not 3. Our molecular data support 2 traditional subfamilies, the plumose-seeded Tillandsioids and the fleshy-fruited Bromelioids, as being monophyletic. By contrast, the traditional Pitcairneoids, marked here by the syncopated green bar, is paraphyletic, and we had to divide it into 6 subfamilies to maintain the monophyly of each. Within the Bromeliaceae, the remarkable genus Brocchinia forms the subfamily Brocchineoidea, a small clade of 20 species sister to all other bromeliads. Brocchinia is endemic to the Guyana Shield in northern South America and is nearly restricted to extremely nutrient-poor soils that are atop the tepuis and adjacent sand plains. In response to extreme nutrient poverty, Brocchinia has undergone adaptive radiation in mechanisms of nutrient capture that's unparalleled in angiosperms, including carnivores, ant-fed myrmecophytes, tank epiphytes, and nitrogen fixers, in addition to terrestrial rooted species ranging from tiny lithophytes to gigantic tree-like forms second only to some of the puyas in size. Lindmanioidea is the next rung on the bromeliad ladder and includes 2 terrestrial genera restricted to the Guyana Shield. Tillandsioidea, the subfamily most specialized for epiphytic life, and including 1/3 of all bromeliads, diverge next. Most Tillandsioids occur in cloud forests in the Andes, Central America, and the Caribbean. Hectia, spinous and succulent, and grows in arid habitats in Central America, Mexico, and Texas. Navioideae, sister to the remaining bromeliads, is restricted to the Guyana and the Brazilian shields. All are terrestrials with tough, non-impounding leaves. Navia is the most species-rich genus in the Guyana shield, but is ecologically very stereotyped. Its diversity appears driven not by adaptive radiation, but simply by poor seed dispersal. Its unwinged seeds have no means of long-distance dispersal. The recircumscribed Pitcairneoideae include 5 genera. Pitcairnea itself grows from sea level to the Andean crest, mainly in forest understories, and is marked by broadleaves and showy flowers, pollinated by hummingbirds. Fosterella occurs at mid-elevations, and Deutericonia occurs as a cushion plant at the highest elevations, where hummingbirds will actually hop from one flower to the next to pollinate them. Dickia ranges from the Andes to the Brazilian shield, and then overlaps with Encolurium that extends into the arid Horn of Brazil. Puya is centered in the Andes, and several taxa bear spectacular iridescent flowers and immense terminal inflorescences. Jubalia and Seitzma have shown that Puya consists of 2 clades, one mostly in the high Andes, and the second from lower elevations in coastal Chile. Finally, the fleshy-fruited bromelioids are sister to Puya. The earliest divergent bromelioids are all small terrestrial genera, including 3 from low elevations in southern Chile, Bromelia from throughout South America, and Dinacanthon pseudonanus and the pineapple genus Ananas from the Brazilian shield. Most of the bromeliads, however, are tank epiphytes native to the Atlantic rainforests of southeastern Brazil. As you can see here, we calibrated our bromeliad phylogeny against the age of 6 monocot phylogenies, attained a stem age of 98 million years, and a crown age of 23 million years. So a lot of extinction in this group. And also this big gap between when the group diverged from other monocots and when they began diversifying helps explain why morphologically bromeliads are so divergent from other monocots. We use this phylogeny to analyze geographic spread using maximum parsimony, which shown here as the branch colors, Bayesian inference, shown as the colors of the pi diagrams at each node, and S-diva, shown as the larger pi diagrams. And as you can see, the conclusions we reached using these 3 approaches are essentially the same. Our phylogeny implies that the bromeliads arose in the Guyana shield, coded in dark blue, and began spreading outside this region 18 million years ago into the Andes, Central America, the northern South American coast, and the Caribbean. 2 major invasions of the Andes, coded in red by Tillandsioids and by Pitcairneoids through bromelioids, began 15 million years ago. Hectia invaded Central America, coded in yellow, no later than 12 million years ago. 2 major invasions of the Brazilian shield, coded in light blue, began 10 and 5.7 million years ago, with Dickia and Encolurium in the semi-arid interior, and the core bromelioids in the Atlantic forest region along the coast. Pineapples in the genus Ananas evolved between 8 and 2 million years ago. Tragically, it was not until much later, in 1955, that pineapples finally morphed into pina coladas, their ultimate and optimal form. We envision the interplay of bromeliad adaptation, distribution, and diversification as follows. First, fertile montane habitats favor epiphytes by creating high rainfall, low evaporation, and a rich rain of nutrients. Second, epiphytism should favor tanks, absorptive trichomes, and CAM photosynthesis to provide and conserve water and nutrients while the plants lack contact with the soil. Tanks should aid moisture and nutrient capture in fertile, rainy, montane habitats. And tanks, absorptive trichomes, and CAM should favor epiphytism. Arid terrestrial sites, however, should also favor CAM, and CAM should promote the invasion of such sites as well. Third, epiphytism should select for entangling seeds, that is to say, seeds in fleshy fruits or with fine plumes that facilitate attachment to twigs and branches, either through birds regurgitating the seeds or through the plumes wetting and clinging to those twigs and branches. Fourth, wet, cool, montane conditions should select for thermoregulating pollinators, including hummingbirds in the neotropics. Fifth, epiphytism should favor high rates of species diversification by providing access to a dynamic and ecologically diverse substrate inhabited by few other plants. Sixth, floral coevolution with rapidly radiating hummingbirds with different bill sizes and shapes should favor high rates of diversification as well. And finally, moderately high dispersal associated with entangling seeds, combined with life in extensive, topographically complex mountain chains, should promote high rates of species diversification, allowing lineages to disperse down such chains and then diverge and speciate in parallel at several points simultaneously. Now, to test all these ideas, we first reconstructed the evolution of key traits, indicated here by branch color, as a function of time over the past 23 million years, indicated by horizontal position on these trees, and by geographic region, indicated by background shading. As shown on the left, there were two major origins of epiphytism in Tillandsioids in the Andes 15 million years ago, and as the northern Andes began to uplift, and in Bromelioids in the Brazilian Shield 5.7 million years ago, as the Serra do Mar and other coastal ranges in Brazil began their uplift. Tank evolution, shown on the right, largely paralleled that of epiphytism. Entangling seeds arose at the same time as epiphytism and tanks in Tillandsioids, but before both in Bromelioids. CAM evolved at least 5 times, mostly in association with arid terrestrial sites, starting 15 million years ago, and associated with Miocene drying and rain shadows caused by Andean uplift. Avian pollination arose twice in ancestors of the Tillandsioids and in the Pitcairneoid through Bromelioid clade, and did so in the Andes at about the same time as their northern uplift and as the two major clades of Andean hummingbirds were arising. Now, using the program Bayes traits, we found significant patterns of correlated evolution for all the pairs of traits predicted by our a priori model. Using the program BAM, and thanks very much, Dr. Ryboski, we found significant patterns of correlated evolution. We found two significant accelerations, of net species diversification in the Tillandsioids, Scotopsis and Glomera pitcairnea, and in the tank-forming Bromelioids and their immediate relatives in the Atlantic forest region. Rates of speciation, net diversification, and extinction in both of these clades are shown in the inset green, red, and black curves. Accelerations of speciation were significantly correlated, according to Bissey, with a number of traits. And specifically, we found, let me just advance this here, that diversification was correlated significantly with epiphytism, with the tank habit, entangling seeds, probably not with entangling seeds, with hummingbird pollination, and especially with life in extensive, nutrient-rich tropical cordilleras, including the Andes and the coastal mountains of southeastern Brazil. The tank epiphytic clade of Brazilian bromeliads had the highest average rate of net diversification. Now, the acceleration of net diversification by particular traits or environments is an important feature underlying bromeliad diversity. But we should also consider the additive effects of broad-scale radiation on diversity. Based on the association of traits with the invasion of particular regions by particular clades, 7 major radiations appear responsible for generating more than 85% of all bromeliad species. First, the rise of primarily C3 epiphytes in tillandsioids, shown in dark green, accounts for about 1,200 species in the Andes, Central America, northern South America, and the Caribbean. Second, the rise of CAM epiphytes in bromelioids, shown in light green, accounts for about 600 species in the mountains of southeastern Brazil. Third, fourth, fifth, and sixth, the origins of CAM terrestrial plants, shown in red, account for roughly 525 species in 4 clades native to arid portions of Central America, the Caribbean, north coast of South America, the Brazilian Shield, and the high Andes. And finally, the rise of broadleaf Pitcairnea, shown in blue, in rain and cloud forest understories, generated nearly 400 species. The 3 clades shown in gold that did not expand outside the ancestral Guyana shield contributed less than 200 species to current diversity. It's often hard to determine which came first, adaptive radiation or species diversification. Do clades evolve more ecological strategies because they evolved more species, or vice versa? If we incorporate the effect of trait evolution on the physiological basis for expansion into new areas, I believe it strongly suggests that the causal arrow flies from adaptive divergence to ecological innovation to invading new regions to species richness. We believe that this kind of approach, based on testing a priori hypotheses regarding function and geographic range, might illuminate evolution in many other groups and help make historical biogeography a hypothesis-driven enterprise. Finally, let's turn to the drivers of orchid diversification. Orchids are the largest family of flowering plants with over 880 genera and 25,000 species. They are more diverse than birds, mammals, and reptiles combined and grow in a wide range of habitats worldwide. Essentially, all temperate orchids are terrestrial, but most orchids inhabit tropical forests, and over 80% of those are epiphytic. Orchids display remarkable floral diversity, and sexual selection run rampant, with striking adaptations to different pollinators and partitioning of individual pollinators by precise placement of pollen packets or pollinia on different parts of their bodies. The evolution of pollinia, specialization on individual pollinators, pollination via deceit, or euglossine bees, or Lepidoptera, epiphytism per se, or associated traits like CAM photosynthesis, and distribution in the tropics, and especially in extensive Cordilleras, have all been proposed as drivers for the extraordinary diversification of orchids. Tests of these hypotheses have, however, been stymied by the absence of a fully resolved backbone phylogeny. Resolving relationships within the subfamily Epidendroideae, including 80% of orchid species, has proven particularly problematic. So to clarify orchid phylogeny and test theories about the impact of various traits on diversification, we amassed data for 75 plastid genes from 39 species representing all orchid subfamilies and 16 of the 17 tribes, as well as 100 outgroup taxa. Maximum likelihood produced this fully resolved orchid phylogeny, with 2/3 of the nodes having bootstrap support greater than 97%. This plastome tree clarifies broad-scale relationships within the family at several levels and resolved relationships among almost all tribes of the epidendroids. Now, to produce an even more finely resolved phylogeny, we conducted a supermatrix analysis where we added 144 additional species represented solely by sequences for 3 plastid genes. And then we calibrated this supermatrix tree against time using beast and 17 angiosperm fossils. The result that's summarized on the right to compare with the previous views of phylogeny is a fully resolved, time-calibrated tree for the orchids and a major advance on that previous consensus view of orchid phylogeny. Orchids appear to have diverged from their common ancestor of other asparagales 112 million years ago and began diverging from each other 90 million years ago. The 8 tribes and 3 previously unplaced subtribes of the upper epidendroids diverged rapidly from each other between 38 and 31 million years ago. Short branches also separate 4 of the 5 tribes of the lower epidendroids. Based on a BAM analysis the rates of speciation and extinction at the Asparagales crown were 0.070 species per million years and 0.016 species per million years, respectively. We found three significant accelerations of net diversification within the order and one significant deceleration, all of them within the orchids. One acceleration occurred at the base of the orchidoids, and the three remaining shifts are nested within the upper epidendroids. Warmer colors indicate higher rates of net species diversification. So diversification accelerated at the base of the upper epidendroids, decelerated in Calypsoe through Blattiani, and then maximally accelerated in the core epidendria. We'll get to the biology of this in just a moment. The initial rate of species diversification in the latter group, limited mostly to the Andes, was more than 6 times that at the crown of the Asparagales. We overlaid traits thought to influence diversification on the supermatrix tree and then used BISSI to test whether they were correlated with speciation and extinction rates with increased net diversification within lineages or with increased net diversification across lineages via added origins or losses of the trait in question. Here, lambda 1 and lambda 0 are speciation rates with and without the character state in question. Mu 1 and mu 0, the corresponding extinction rates. Zeta is the estimated advantage in net diversification per million years within lineages conferred by a character state. Q01 and Q10 are the rates at which the focal character state is gained or lost per taxon. And Z is the estimated advantage in net diversification across lineages conferred by a trait. The evolution of Pollinia significantly increased speciation and extinction and resulted in a 5.1% advantage in net diversification per million years within lineages. Lest you think that is small, remember it's compound interest. So in 20 million years, that's a lot of difference in diversity. Pollinia should accelerate speciation in our view by facilitating specialization on individual pollinators or parts thereof, and by reducing the number of independent matings per population. Pollinia may also allow small numbers of variants to produce numerous offspring, promoting speciation from small numerical bases that might otherwise lead to demographic collapse and extinction. Epiphytism significantly accelerated both speciation and extinction rates relative to the terrestrial habit, increasing both fivefold or more and yielding an 8.8% advantage in net diversification per million years within lineages. Epiphytism is a key innovation that allows the invasion of a new adaptive zone largely unoccupied by other vascular plants. Epiphytism should also help generate and maintain high levels of plant diversity because the habitable surface of bark, branches, and twigs is much greater than the ground occupied by a forest. Because variation in sunlight and humidity within tree crowns permits local niche partitioning, and because altitudinal and topographic variation across mountainsides can be partitioned at broader scales and foster local speciation. Epiphytism is associated with high rainfall and humidity, and thus with tropical mountains. Tropical distributions appear to accelerate speciation and extinction, but this difference appears driven primarily by habit, not latitude. We found that terrestrial lineages in and outside the tropics had essentially identical diversification rates, while tropical epiphytic lineages diversified 2.3 times faster. CAM photosynthesis accelerated speciation and extinction rates, though not significantly, yielding a large advantage, however, in net diversification within lineages and a smaller disadvantage across lineages. In orchids, CAM is strongly associated with epiphytism. Deceit pollination via mimicry of food sources, nesting sites, or potential mates is nearly restricted to orchids. It significantly increased speciation and extinction rates, but resulted in little overall change in the rate of net diversification. Mimicry of potential mates should accelerate speciation because subtle shifts in flower form or scent could attract different pollinators and lead to reproductive isolation. Pollination by male euglossine bees had no significant impact on speciation or extinction, but increased net diversification by 3.6% overall. We'd expect euglossine pollination to accelerate speciation by allowing small chemical changes to attract different bee species. Pollination by Lepidoptera doubled speciation and extinction rates, within lineages, resulting in a 6.9% acceleration of net diversification per million years overall. Pollination by moths or butterflies involves nectar presentation in corolla tubes or spurs whose lengths might rapidly evolve and lead to the rapid recruitment or evolution of new pollinators and thus enhance diversity. Tropical, life in tropical cordilleras, specifically the Andes and the New Guinea highlands, significantly increased speciation and extinction rates, yielding a very large 25% advantage in net diversification rates within lineages, but also apparently a large disadvantage across lineages. But the latter is greatly overstated because most lineages never invaded those cordilleras and thus can't contribute to the cross-lineage disadvantage. Extensive tropical cordilleras thus appear to be the strongest overall effect on net diversification in orchids. To summarize, we've shown that, let's just move on to this here, that orchids are remarkably species-rich, partly as a result of 3 accelerations of net diversification. Our data provide the first quantitative support for several hypotheses regarding the genesis of orchid diversity and identify specific points in orchid evolution where these factors appear to have played a role. They show that multiple factors, several of them interconnected, have contributed to orchid diversification. The first significant acceleration of net diversification is at the base of the orchidoids, one node above where Pollinia evolved. The second acceleration coincides with the origin of epiphytism in the upper epidendroids and the rise of several montane clades. The deceleration within the upper epidendroids, shown by those blue branches, reflects the evolution of a grade that is composed entirely of temperate terrestrials and of lowland epiphytes. And then finally, the largest acceleration in the tribe Epidendriae coincides with a large concentration of Andean taxa pollinated by deceit or by Lepidoptera. The pleurothalids, the most diverse component of this clade, are largely pollinated by flies, lured by deceit. Low diversification rates persisted in the lower epidendroids, which are terrestrial and essentially lack pollination via deceit, euglossines, or Lepidoptera. Overall, our results show that epiphytism and life in extensive tropical cordilleras, as well as pollination by Lepidoptera and euglossine bees, have had large effects on orchid diversification. The defining characteristics of orchids, namely tiny seeds and mycoheterotrophic germination and the floral column, did not themselves accelerate diversification of orchids, but appear to have facilitated this diversification driven by the evolution of Pollinia and epiphytism. To conclude, though, this is not the entire story. Several factors implicated in orchid diversification also helped generate the diversity in Bromeliaceae, but bromeliads evolved fewer than 3,300 species. Why do orchids have nearly 8 times as many species? I think there are 4 factors that might help account for this difference. First involves dispersal, time, and familial age. The dust-like seeds of orchids, their ability to grow at a wide range of latitudes, and the much greater time since their initial diversification, 90 versus 23 million years for bromeliads, almost surely fostered greater diversity by permitting orchids to spread to every continent and latitude, or most latitudes, I should say, while bromeliads remained in tropical and subtropical America with the exception of 1 species. Across flowering plants, families with broader geographic and latitudinal ranges are more diverse. Our preliminary analysis of historical biogeography, which I'm not going to show today, indicates the orchids arose in South America, Australia, and yes, Antarctica, 112 million years ago. And repeated intercontinental dispersal appears to have at least quintupled orchid diversity beyond what it would have been without dispersal from their original continental range. Second point, karyotypic evolution. Chromosomal evolution is often overlooked as a diversifying influence in orchids, but many of the large orchid genera show extensive polyploid and aneuploid variation in chromosome number. Pollinia and tiny seeds allow recovery from repeated population bottlenecks Such bottlenecks could fix karyotypic mutants via drift and quickly generate post-mating barriers to crossing. Third, limited dispersal ability of pollinators. The evolution of dozens of closely related species of Tegia, for example, only a few kilometers apart in Andean Ecuador, and more generally the rapid geographic turnover of epiphytic orchid species with distance in the Andes are both consistent with gene flow over only short distances in tropical montane areas. More research is needed to determine whether that is a result of perhaps a rapid rainout of seeds due to the heavy rainfall in those areas, or limited physiological tolerance or dispersal ability of the small, soft-bodied, weak-flying, desiccation-intolerant flies that pollinate many of the orchids at that level. Finally, greater species packing. Across 17 neotropical forests surveyed by Kreft, epiphytic orchids had 5.6 times as many species within stands as bromeliads, and more geographic turnover from site to site. Higher levels of local diversity suggest that community assembly in orchids may involve a finer or more extensive partitioning of pollinators or occupation of a wider range of perches than in bromeliads. Higher levels of spatial turnover suggest that despite their dust-like seeds, many epiphytic orchids or their pollinators actually have quite limited dispersal capabilities, perhaps reflecting limited dispersal by their pollinators or mycorrhizae. These possibilities call for a lot of new research on orchid and bromeliad community assembly, on the physiology and dispersal abilities of montane dipteran pollinators, and on the scales of spatial genetic structure and gene flow in epiphytic orchids. More comparisons between lineages, like the ones I've made here today, between orchids and bromeliads will also be needed to see whether hypothetical drivers of diversification are truly adequate to account for the observed differences we see as we begin to explore the determinants of lineage size across all monocots. Finally, I'd like to thank the National Science Foundation for its continuous support for my research since 1987, and express my deep gratitude to all of my colleagues for their many contributions to the Monocot project, to the Bromeliad evolution study, and to the investigation of orchid phylogeny and diversification. Thank you. And thank you for your patience. Questions?
I can ask you, because Sarah will be passing around the...
Excellent, okay. All has been revealed, no questions? Yes. My pleasure. In orchids. Yep. Well, partly, if not largely, as a result of Herb Wagner's work, we know that polyploidy and hybridization are not dead ends. They were thought to be, certainly in the 1940s and 50s, but Herb's work clearly showed that hybridization and polyploidy could be creative forces. And in ferns, tremendous amount of diversity has arisen as a consequence of those processes. In Herb's day and in Ledger Stebbins' day, people debated what fraction of all flowering plants were the result of polyploidy. And they came up with various numbers, 20% or 40% or maybe 45%, and even higher, of course, for the ferns. Well, it's all rubbish because, in fact, essentially everything is an ancient polyploid as a result of genomic studies we can see there have been multiple gene duplication events over time. And if polyploidy is a dead end, we're surrounded by them. Yes. Yes. Many people have never seen them. Yes. In many cases, yes. I mean, one of the questions, of course, is why has mycoheterotrophy arisen so frequently in monocots as opposed to other lineages? So there are lots of cases in the monocots and very few outside the monocots. And you know, we've thought about this a fair amount, and I think one of the considerations probably has to do with the fact that the infection of the mycorrhizae associated with primary root production. And pardon me, monocots don't have secondary root thickening and so on. So almost all the roots they have are primary roots, and so they are especially likely to get infected. It's also pretty clear that mycoheterotrophy is associated with shady environments in many cases, where the plants may not be giving up much in terms of photosynthesis to turn to the dark side and start stealing sugar from their erstwhile fungal mutualists. But it's a very common strategy in just one family, really, the Bromeliaceae, but has arisen in many different monocot families. So there is even a mycoheterotrophic iris. And mycoheterotrophy has arisen at least 30 times within the orchids alone. So now in the orchids, probably that happened because, as with Botrychium among the ferns, there's an obligate heterotrophic stage early in life where the seeds of orchids only germinate as a result of contact with mycorrhizal fungi. They get food from the mycorrhizal fungi. In Botrychium, the plants live underground for a year or two without getting carbon from photosynthesis. Presumably, in both cases, the evolution of complete mycoheterotrophy has been facilitated because the plant depends on it for part of the life cycle. And you have things like the goblin fern among Botrychiums that is essentially mycoheterotrophic. And as I said, over 30 origins of mycoheterotrophy. Now, just to conclude that thought, though, mycoheterotrophy is never associated with a lot of species. In other words, the lineages involved are fairly small. So it is not it doesn't add a lot to orchid diversity. It adds a lot functionally, and the plants often look really weird, but it doesn't add very